Hi, welcome to another lecture. This lecture will be about connective tissue once again. I hope you have a nice day. Today we will focus on another type of connective tissue, namely cartilage and bone. So let's start. Connective tissue, as you remember, hopefully, I hope so, consists of cells and intercellular mass or extracellular matrix. That's just another name for intercellular mass. And this mass predominates in connective tissue. But cells are essential for the formation and maintenance of the matrix that is around them. So they are very important, and we distinguish two types. And we distinguish resident or fixed cells and migratory or wandering cells. Fixed cells are of mesenchymal origin. All fixed cells arise from undifferentiated mesenchymal cells here. Really fixed cells are permanent inhabitants of the connective tissue and form the mass. They produce the mass. Today we will be interested in chondrocytes and chondroblasts, which are their precursors. So chondroblasts are precursors of chondrocyte here. These guys are in cartilage. Next ones are osteocytes and osteoblasts, and once again this is the same. Osteoblasts are precursors of osteocytes, and these are in the bone. So these are the main fixed cells in the cartilage and in the bone right now. Another type are migratory cells or wandering cells, which are these cells are derived from hematopoietic stem cells. So we have some stem cells in the bone marrow, and these migratory cells are derived from these stem cells. Do you remember in the proper connective tissue we had mast cells or plasma cells, which differentiate from B lymphocytes, and also macrophages, free macrophages? So in bone we can find osteoclasts. Normally you have many osteoclasts in the bone, resorbing the bone, transforming it. So osteoclasts are normal migratory cells in the bone. Then we have chondrocytes also. So we have osteoclasts in the bone, and we have also chondroclasts there. But these guys are a little bit different. They're not normally located in a cartilage all the time. There is just one specific process where we can find chondroclasts. So just remember right now, osteoclasts are normal migratory cells in the bone. So once again, intercellular mass is composed of two components, and that is a fibrillar component and amorphous component. The fibrillar component consists of fibrils, and these fibrils are formed by collagen, or we can rarely find elastic fibers, in the bone, and not in the bone, in the cartilage. So just remember collagen. This is the most important right now. Fibrillar component is made of collagen, and there are various types. And remember, only some types of collagen form fibers. Do you remember collagen type 1? This type of collagen is forming fibers, of course. Collagen type 2 doesn't form fibers, and that will be very important and crucial in differentiating many types of cartilage, for example. And there is this amorphous component. The amorphous component is composed of low molecular weight substances such as ions and small molecules, but don't forget about water. Water is also very important. So amorphous component right here. But so are we have also macromolecules that are in an amorphous component here, and these macromolecules are these glycosaminoglycans and proteoglycans and structural glycoproteins. Glycoproteins. Yeah, so these are the macromolecules which are forming amorphous component. But don't remember water and ions and another smaller molecule that are also in there. So now specifically for each type, yeah, so first is cartilage. Cartilage. So we have some resident cells in cartilage, fixed cells, and these guys are called chondroblasts, which are precursors to the chondrocytes. Do you remember fibroblasts and fibrocytes in proper connective tissue? I hope so. These fibroblasts were able to divide and were more active than fibrocytes. Yeah, fibroblasts have a lighter, more active nucleus, and fibrocytes are not able to divide and they're not so active in the terms of production and stuff. And here we have chondroblasts and chondrocytes, and these are there's some difference between these because in comparison to fibroblasts and fibrocytes, both of these are able to divide. So they can undergo mitosis. Both chondroblasts can divide and also chondrocytes can divide. So that's a little bit different. And even though chondrocytes are less active, yeah, that's the same. They are less active than chondroblasts, but they are so that active they can divide. Yeah, I hope you understand. And as a result that these can divide, they form isogenous groups in the cartilage. Isogenous groups. Yeah, they can form these groups. And I will show you these groups, how do they look like, but it's really, there are many cells in one place like this, and this is because of their division, you know. So yes, and then we have these migratory cells also in cartilage, but these do not occur commonly. As I said before, in cartilage, that's not normal. You have chondroclasts in cartilage. They are there only in a certain process. These cells really resemble bone-like osteoclasts. So they're really just the same things, osteoclasts. But they are specialized, these chondroclasts are specialized for the cartilage, for the resorption of cartilage. And this happens only during cartilage remodeling, in the process of bone formation. So when you want to form a bone, you need this cartilage model first. Then you resorb this model or completely destroy it, and you form bone in that place. So we're going to explain these today also, the formation of bone and this stuff, which are very difficult. But yeah, we get there. But just remember right now, chondroclasts, we are not able to find them normally in a cartilage, but only in this process of bone formation, when you want to get rid of cartilage that's there. Yeah, so it's not normal that we are not able to find them normally. The fibrillar component is essential in distinguishing cartilage types. So we have three basic types of cartilage. And when you want to distinguish these types, you look for the fibrillar component of extracellular matrix. But remember one thing, we always find type 2 collagen in cartilage. That's very important. Type 2 collagen is crucial for cartilage formation, and you always find this collagen type 2 there. This collagen forms fibrils. And these fibrils are so thin that we can't really see them even in electron microscope. It's really hard to see them there because they're so thin. Only very, very high magnification can show us fibrils. So remember, very thin fibrils, yeah. And collagen type 2 can't form fibers, so no fibers. No fibers, really no fibers. But there are types of cartilage that have fibers, yeah? So some cartilage doesn't contain fibers, but some cartilage or some types of cartilage do contain fibers. And for example, that's for the fibrous fibrocartilage right here. Fibrocartilage also contain collagen fibers, and these are type 1 collagen fibers, yeah? When we talk about collagen fibers, we really mean type 1 collagen. Or elastic cartilage, which contains, well, elastic fibers, yeah? So that's obvious, I guess. Elastic fibers in elastic cartilage, collagen fibers in fibrocartilage, yeah? But these types of cartilage also contain type 2 collagen, always, yeah? So don't forget about type 2 collagen, very important. Amorphous component of the intercellular mass are aggregates composed of hyaluronic acid and proteoglycans containing chondroitin sulfate and keratin sulfate. Yeah? Chondronectin is example of structural glycoprotein. Chondronectin. Yeah, remember these structural glycoproteins connecting everything together. Yeah. So structural glycoprotein and these glycosaminoglycans and proteoglycans are forming these very big aggregates in the cartilage. Really, the cartilage has such a firm consistency because of these that it does not let blood vessels inside to go in, yeah? So the cells in the cartilage are nourished only by diffusion because of this amorphous and fibrillar component in the cartilage. It's very firm consistency, and really no vessels can penetrate through this stuff that's there. So only diffusion in the cartilage. This is very crucial because this also limits the size of the cartilage and also healing of the cartilage. So just remember, cartilage heals with a fibrous scar. Yeah, it's not able to heal or regenerate. Once you destroy your cartilage, it's destroyed. End of story. Yeah? No regeneration. So yeah. So this is just a picture. Just a drawing, but it's very nice. I like it. We can see here that on the cartilage surface there is usually a layer of dense irregular connective tissue. Remember, dense irregular connective tissue, and that's called perichondrium. This is the layer of dense irregular connective tissue on the surface. Yeah. It's the last tissue, the perichondrium. It's the last tissue that contains blood vessels. So we are not able to find blood vessels in the cartilage, but perichondrium contains blood vessels. So really the perichondrium nourishes cartilage, and also it contains nerves. Yeah, so also nerves are here. It's made of dense irregular connective tissue once again, and this really means that perichondrium is formed of type 1 collagen. Yeah, so we can find collagen fibers there, and it's type 1 collagen. And really there is a cartilage between, so here we have this perichondrium. Yeah, there are fibroblasts because it's just collagen tissue, so there are fibroblasts there. So type 1 collagen. And then we have this cartilage, and this is an example of hyaline cartilage, the first type. And we can find only collagen type 2 here in the cartilage itself. And remember, type 2 collagen doesn't form fibers. So here we can find fibers in the perichondrium, and we are not able to find fibers of collagen type 2 because simply they don't exist. Yeah. So you can't describe fibers during oral exam, for example, because they don't exist. Yeah, type 2 collagen fibers, really, that's not what's happening here. Only fibrils, and we can see them only in electron microscopy. So that's very, very important. So once again, we can see the surface area of cartilage here. We have these flattened cells there in the surface area, and these are called chondroblasts. Yes, so in the surface here, there are chondroblasts. Around them, there is an extracellular matrix that is formed by chondroblasts, by them. So around here we have extracellular matrix. It's not so pretty and mature. It does not contain so many aggregates, so it's kind of loose here, this matrix. And we can see, because of that, the chondroblasts are individually really undergoing mitosis and separating from each other. So these chondroblasts are dividing, but because this matrix that is around is so loose, there are not so many aggregates there, they can simply go and to another place and be alone there, just sit there, so they can simply migrate somewhere else. Yeah? And they are located in lacunes. So they are in lacunes. For example, like this, there is a lacune, like a house for this cell. And each cell has its own lacune right here, space around, because it needs space. The further we go, the more dense the matter is, really. So here is really the denser matter than here on the surface. And cells are more round. We can see this spherical cell right here. It's more round. And they really accumulate fat and glycogen inclusions. And these are called chondrocytes. So they are more spherical and they accumulate glycogen and lipids right here. Yeah, and why do they do so? Why do they accumulate lipids and glycogen? It's because there is not so good nourishment here. There are no vessels, so they want to store something. Yeah, because they don't want to die of starvation, for example. Yeah, so they are storing something. So they are storing lipids and glycogens. These chondrocytes were formed by mitotic division, and we can see it right here. We have probably one mother cell here. For example, this chondroblast right here was divided into two cells, these two chondroblasts, and they stayed together because this matrix that is around is so dense that they were not able to move from this place. Yeah, and they undergo mitosis even more right here. So they are together here in one lacune, here in one space, and this is an isogenous group right here. But this was just the picture, and here we have a picture from light microscopy. So once again, this is a perichondrium. Dense irregular connective tissue. We can really see collagen fibers. This is a collagen type 1, yeah, making those fibers. It looks kind of regular, but we can also see cross sections here, so it's not that regular as tendon. It's still irregular connective tissue, this perichondrium. Here on the surface, right here, we can see these narrow cells in their own vacuoles, undergoing mitosis probably. Yeah, and these are chondroblasts. Chondroblasts right here. Here we can see more spherical cells right here. One cell here, one cell here, another one here. Sometimes we are not able to see nucleus. Nucleus is this dense thing right here. And they are forming isogenous group right here. Or these guys right here are forming isogenous groups together. And don't forget, chondrocytes are able to divide also. And that's why there's so many of them in this one place right here. We can see, this is an example of the first type of cartilage. This is a hyaline cartilage. And we can see only some eosinophilic matter right here. It's kind of homogeneous. There are some denser places, or denser I should say, there are places here that are more eosinophilic, some of them are less eosinophilic here, but we can't really see fibers here. Yeah, we can't see fibers because this is just collagen type 2, which is forming fibrils. Yeah, so no fibers here. And now a little more about the chondrocytes. We can see them here, chondrocytes. Once again, they are spherical cells. Yes, spherical cells. They have these small protrusions that are going out of them, which improve their ability to absorb substances. Yeah, so small protrusions. They are not able to move, so they're just for absorption. These things right here. They live in lacunes. So this is one lacune with one chondrocyte inside. Yeah, so lacune with chondrocyte. And really their nuclei are pressed to the periphery. So for example here, this is a chondrocyte and the nucleus is right here. It's very hard to see, but these nuclei are pressed to the periphery also right here. Yeah, so they are dense, or I mean dark basophilic, and they are placed to the periphery. And once again, the cytoplasm contains lipid droplets. Yeah, those are those bright areas. For example, here is the lipid droplet right here. And also here we have lipid droplets, and here. And the bright areas, they also contain bright areas. Yeah, for example, here we have this bright area here or around here. And these are areas where glycogen is stored. So we have lipid droplets and glycogen. And it's very understandable that they create inclusions. They are afraid that they will be malnourished. Yeah. So they maintain a supply of substances. Yeah, glycogen is very good substance, because you can metabolize very fast glycogen. Yeah, so that's very important. And this is a picture from electron microscope, and we can see that the cytoplasm of chondrocyte contains a lot of granular endoplasmic reticulum. So there's large amount of granular endoplasmic reticulum right here, because chondrocyte is producing, it's a highly active cell. It's not that active as chondroblast, but it's active. Yeah, so it's producing it's also dividing and it's producing. So we have this nucleus right here and granular endoplasmic reticulum, and we have mitochondria there. They're always in the cell, mitochondria. And these dots that are around, these things right here, these are cross sections of type 2 collagen fibrils. Yeah, so we have fibrils here around, fibrils. And also let's not forget that it produces an amorphous component that forms aggregates. Yeah, so not only collagen type 2, but also this amorphous component is very important in cartilage. Yeah, once again, this is a picture from hyaline cartilage, and these are isogenous groups right here, isogenous groups. So this, yeah, and this is probably together one isogenous group, and also here. So we see isogenous groups in many places here. Here we have, on the surface, we have these chondroblasts, yeah, and these are chondrocytes deeper in the cartilage. So this is a name picture from one isogenous group, yeah, and the intercellular mass is basically eosinophilic due to collagen. We can see it here. It's basically eosinophilic. It's pink because of the collagen that is there, but it's divided into several areas here, and the degree of eosinophilia varies. Yeah, some places are also even more basophilic. Yeah, so in general there is eosinophilia around, but we have this capsular matrix, yeah, that is around a lacune right here, and this capsular matrix or capsule in general is more basophilic. Yeah, it's basophilic. It's sometimes called also a basophilic capsule because of that. So this matrix that is close, very close to the lacune is basophilic, and it's called basophilic capsule. And right next to these basophilic capsules, yeah, right next next to these chondrocytes that are forming isogenous groups, we have this matrix. And because it's in the territorium of these cells, it's called territorial matrix right here. The matrix that is close to the chondrocytes is called territorial matrix. And really between territorial matrices of several isogenous groups, we have interterritorial matrix. So this is the matrix between territorial matrices. I will show you that on the picture from light microscope, because this is very not a good picture. And this is also very not good picture, but okay, this is very dark here. I'm having another picture? No, I'm not. I will explain it here. Maybe it will be better. So what I wanted to say is that so here we have this picture. We can see these chondrocytes in forming isogenous groups here. You can see those. And you can see this slightly basophilia around. I hope you can, right here. And all around each lacune, this is the basophilic capsule or capsular matrix that is around. Capsular matrix. It's basophilic capsule or sheath. Furthermore, the matrix, which is weakly eosinophilic, forms a filling between chondrocytes, yeah, within the isogenetic groups. So we have filling here between chondrocytes and here around, and this is territorial matrix that is here also between chondrocytes and like this all around. A little further from the isogenous group is interterritorial matrix, and it's a little further, and it's right here, like this, or here we can see it like this. It's darker, or here. So we can see there is lighter eosinophilic matrix that's here, and then we have this darker matrix that is between isogenous groups right here. So first there is this basophilic capsule around, and then there is territorial matrix that is lighter, and after that we have interterritorial matrix that is darker. And the reason why it's darker, the reason why we can see these darker places right here, is that interterritorial matrix is more mature. It contains more type 2 collagen, so it's more eosinophilic because it contains more type 2 collagen, and we know collagen is eosinophilic. So when there is more collagen, we have darker eosinophilia because of the collagen. So and this is the picture of a cartilage from higher magnification, very high, and you can see it's very dark. So probably it's some kind of different staining, but once again we can see it, and we can see it even better right here, because here we have this isogenous group that contains 3 chondrocytes, probably. We can see it in this section right here, and these chondrocytes right here, each of them contains capsule. Oopsie, I'm so sorry. So there is a basophilic capsule around. Yeah. So there is a basophilic capsule, and then we have this territorial matrix that is lighter. It's not so mature as interterritorial matrix that is darker. But what we do have is more collagen type 2 here in this interterritorial matrix. On the other hand, territorial matrix contains more glycosaminoglycans, yeah, but less collagen type 2. So that's it basically, and you should know this. That's why I'm repeating it many times. So, and for the amorphous component of intercellular mass, the basic proteoglycan is aggregan. Aggregan is basic proteoglycan. I'm going to write it down. Aggregan. We can see the monomers. Yeah, we can see a monomer of aggregan right here. Its axis is formed by protein. Yeah, so this is some kind of protein, and these brushes, which protrude from the protein carrier to the sides, yeah, these brushes are glycosaminoglycans right here. So these brushes are glycosaminoglycans. Oh, it's written here. Yeah, it's a protein core, glycosaminoglycans. This is a proteoglycan, and the typical proteoglycan for cartilage is aggregan. It's very typical. And what we can see here, really each aggrecan is attached to a long molecule of hyaluronic acid, or simply called hyaluronan. Yes, so proteoglycans are attached to these hyaluronic acid, and you can see there is some kind of link protein here, but don't worry about that. Link protein here. And really hyaluronic acid itself doesn't need to bind to the protein carrier. Yes, so there's no need to bind it to some protein carrier, and individual aggregants are linked to it. Yeah, you can see it right here, like these interdental brushes, really attached to the hyaluronic acid, and it's connected together. And these eosinophilic structures here are collagen 2 fibrils, and you can see these structures are also connected somehow to the fibrils. Yeah, so everything is interconnected together. And yeah, that's how it looks like, and it's very dense. It's hard to migrate through cartilage because it's very stiff and firm. And just remember this aggrecan, that is a proteoglycan, this interdental brush with glycosaminoglycans on the protein carrier, and the hyaluronic acid is very important here. And everything is connected to the fibrils, collagen type 2 fibrils. And what's the macromolecule that is interconnecting these two, the collagen and the proteoglycans? Well, the structural glycoproteins like chondronectin. Yeah, so also remember this chondronectin that is connecting everything together. This is how it looks like. Yeah. So really what we can see here, probably somewhere here, is the protein core, and here should be those brushes from glycosaminoglycans. So this is just one proteoglycan that is attached to the hyaluronic acid right here. And here we have the hyaline cartilage, the first cartilage. So now let's have a look on these three basic types. Yeah, hyaline cartilage, elastic cartilage, and fibrocartilage. Hyaline cartilage occurs most often. Through development, it forms a primary skeleton in the development that transforms into bone. So it's very important in bone formation, also the cartilage. And it's located under articular surfaces, so surfaces in the joints, in epiphyseal plates also, so you can grow. And it's also in the airways, in the nose. It also forms the ventral ends of the ribs. Yeah, normally it has such a white, glassy appearance, and this really means hyaline, that it's like a glass. Yeah, so that's why it's called hyaline cartilage. And its chondrocytes form isogenous groups. So here we can see these isogenous groups right here. Yeah, some of them are small, containing only 2 chondrocytes or 3 chondrocytes, like here. Some of them are bigger. Yeah, there can be up to 7 to 8 cells there in the groups, and these groups are quite far apart. Yeah, so they're quite far apart. There is this space in between, and there is a lot of interterritorial matrix. So this space here, this here is interterritorial matrix between. So once again, we can see these light places right here that are really next to the chondrocytes. This is territorial matrix, lighter eosinophilic, and then we have this dark eosinophilic or darker eosinophilic place here, and this is interterritorial matrix. Yeah, sometimes it's hard to tell in the cartilage. Sometimes you can't really see the difference, but here I think here we can see it quite good in this photo of a cartilage. And remember, there is only type 2 collagen. Type 2 collagen. Yeah, that's very important. So we are not able to see fibers. There is just this like mass here, but no fibers. So next, this is a photo from electron microscope. Yeah, these are chondrocytes. This is chondrocyte, and chondrocyte over here. And we have a little bit of chondrocyte. So there are just chondrocytes here. They contain a lot of granular endoplasmic reticulum because they are actively producing. Yeah. And they also contain glycogen. Yeah. So, and we can't really see many collagen fibrils here because they are in this territorial matrix that it's not so matured, and there is just most of it is formed by amorphous component here. So there are just some small fibrils of collagen here, probably here, but there is more of an amorphous component here, so it's not so mature, this mass around. Oh, and here we have staining with Alcian blue here, and this is a different situation because Alcian blue is used for the staining of sulfatate glycosaminoglycans. So it stains glycosaminoglycans, and we know that territorial matrix contains the matrix that is around the territorial matrix here contains a lot of glycosaminoglycans. So that's why it's stained very well with Alcian blue. So it's like reversed than from the previous slide when we used hematoxylin and eosine. The interterritorial matrix was darker. And here in Alcian blue staining, it's opposite because interterritorial matrix here is lighter because it contains smaller amount of glycosaminoglycans. Yeah, so it's reversed, it's opposite. Next we have elastic cartilage, which is different from hyaline cartilage. And elastic cartilage, really, what we can see here in this picture, there is a perichondrium. Once again, we can see this dense irregular connective tissue, perichondrium. Here we have first cells right here, and these are the chondroblasts right here. And these are the round spherical cells right here, are chondrocytes right here. We can find elastic cartilage. There is this help, like 3 E's. Yeah, we can find it in external ear. So the auricle right here, this structure, you can touch it right now and feel how it's elastic. You can form some things from it. Oh, it's painful a little bit, but okay. So the external ear. Then we have a Eustachian tube. I don't know how to pronounce it correctly, but I'm going to write it so we can see it. Eustachian tube, yeah, that is connecting nasopharynx with the middle ear, and epiglottis. So 3 E's. Epiglottis right here. Plus, there are some smaller elastic cartilages in the larynx, also in airways. But these 3 are the most important for us in histology. And So really why it's elastic, because it contains also collagen type 2, that's crucial for cartilage, collagen type 2, but also it contains elastic fibers, and we can see elastic fibers here because there is no longer these homogeneous matrix around with no fibers, but here we can see these lines right here, and these guys, these are the elastic fibers right here. I can tell you because, yeah, we know that. These are elastic fibers because they are thin. They are not so very dark eosinophilic. There's more light eosinophilic. But you will be able to see that during practical trainings. They are very shiny. They're shining like stars. No, not actually, but these are the shining fibers right here between chondrocytes. Yes, so these are elastic fibers, and that's why it's called elastic cartilage, really, and because it's elastic. You're not able to do this with hyaline cartilage. No can do. Yeah, so only with elastic cartilage. Here, I probably have red ear right now, so I'm going to cover it. Okay. So once again, they do not stain very well with eosin, these elastic fibers, but they stain just a little bit, but they are lighter, yeah. And there is really, when you look at it in general right now, there is less intercellular mass per cell number here. So it looks like there are more cells than intercellular mass. It's not actually like that, but it's different from the hyaline cartilage, as you saw before. So this was the hyaline cartilage. It looks like there is more intercellular mass between chondrocytes, and here in elastic there are more cells per intercellular mass. Yeah, more cells here, and they do not form these big isogenous groups. These isogenous groups right here are smaller. Yeah, sometimes 2, 3, 4 chondrocytes inside, but you can see many lonely chondrocytes right here, lonely chondrocytes. Yeah, so probably elastic cartilage isn't that stiff and firm as hyaline cartilage, and these chondrocytes that undergo mitosis and divide can a little bit move to another place to have this lacune by themselves. Yeah, so they're not this. We're not able to find big isogenous groups here in elastic cartilage because it's more loose. It's not so stiff and firm as hyaline cartilage. And the next we have, this is another picture from elastic cartilage. I think it's the same, nothing special here. Yeah, so you can see here very well these elastic fibers right here. Yeah, you can see these fibers, very thin, light eosinophilic fibers right here. And very special fibrocartilage is the next type. And this fibrocartilage differs, significantly. You can see it. It's somewhere between the cartilage and the dense connective tissue, somewhere between, because there is type 2 collagen, of course. It's cartilage, so we have this type 2 collagen. But we also can see collagen fibers right here, collagen fibers like this. They're going and from the other way like this. And these collagen fibers are, collagen type 1. So there is also collagen type 2 and 1 here. And this one is forming fibers. Two is for the cartilage. Yeah. We're not able to completely define the perichondrium, because perichondrium is dense connective tissue, and also fibrocartilage is somehow like the cartilage, but it's also kind of like dense connective tissue, so you can't very tell the differences. But there is also perichondrium, so it's very gradual transition. And there are chondrocytes also in this cartilage, and we can see that between these highly eosinophilic fibers, very big fibers here, Yeah, so they are collagen type 1 fibers, I can tell you that. So between them, we can see these cells right here, small cells, spherical alone. They do not form isogenous groups or only small isogenous groups with 2 chondrocytes maybe, rarely 3 chondrocytes, but these are chondrocytes. Yeah. So once again, this fibrocartilage probably isn't that firm, or these chondrocytes are not dividing that much, yeah. So they're alone in their lacunes right here. Yeah, there are many of them, quite many of them. Sometimes you're not able to find the chondrocyte in the fibrocartilage, yeah, in some slides. But in this slide, I can see many of them right here. But really, yeah, the ratio is in favor of intercellular mass here. There is more intercellular mass than chondrocytes, yeah, comparing to this elastic cartilage. Yeah, so this is another example, and we can see there are many collagen fibers here, eosinophilic collagen fibers. I should draw them pink, but whatever, I have violet pen here. So collagen fibers, and these cells are chondrocytes, alone, set, chondrocytes right here, because they're alone, and maybe they're happy. I think they're happy because they're alone. Yeah, you should be happy also alone. So I'm alone here right now. mental breakdown. Okay, so let's move on. I'm just making fun. So chondrocytes right here, also from the picture of electron microscopy. I don't know really what's special about this picture. We can see perichondrium once again. Yeah. And I forgot to tell you, where can we find a fibrous cartilage in a human body? Like, where are the places where we can find it? So back to the fibrocartilage. You can find it in a intervertebral discs. Intervertebral discs. The fibrocartilage forms the annulus fibrosus. Yeah, annulus fibrosus in the discs. It's also in the joints in the form of menisci. Yeah, so menisci in the joints and the knee, for example. And also in the jaw, you can find a disc that is from a fibrocartilage. And also fibrocartilage form the symphysis in pubic bones. So also there you can find it. And I'm trying to figure out another place, but I can't. So probably maybe I forgot about something, so you can write it down in the comment section. So let's move on. And electron microscope, yeah, this is a picture. Normal chondrocytes here. This is more, I don't know, probably, no, I said it bad before. This is picture from the fibrocartilage electron microscope. And what we can see here, we can see also collagen fibrils forming a fiber right here. You can see they're going one direction. Probably this is a collagen fiber right here, this structure. And these are chondrocytes normally there. Yeah. And these are probably some collagen type 2 fibrils and also some collagen type 1 fibrils, because we are in the fibrocartilage. And this is a fiber here, this dense thing right here, thick. Yeah, differentiation of cartilage from mesenchyme. Cartilage is formed from the mesenchyme, and it's not so difficult process because we have mesenchyme and then we have a cartilage. So it's not that difficult. And first, mesenchymal cells aggregate in the areas called cartilaginous blastema. So we have some kind of blastema, cartilage blastema, just aggregates of mesenchymal cells. that start to differentiate into chondroblasts. This is how mesenchymal cells looks like. They have these long protrusions and they are like star-shaped cells. And then we have these chondroblasts that are more spherical. They have only very small protrusions on their surface. Yeah, so only very small protrusions, but not these long protrusions like these guys right here. So they lose protrusions here, and they're more spherical around, and they form this blastema right here, blastema, and that means just many cells together, and these are chondroblasts right here. And these blasts start to produce intercellular mass. That's what we can see here. So in this picture we can see we have some intercellular mass in between, and when they do so, they cover themselves with this intercellular mass. They transform themselves into chondrocytes, and here we can see chondrocytes in isogenous groups, and there are many chondrocytes in one isogenous group because they were dividing and they cannot migrate because of the extracellular mass there. So the formation of cartilage is pretty straightforward, I guess. There's nothing difficult about it, but probably the signalization process is more difficult, but not in a histological way. We can see mesenchymal cell, and then we can see a chondroblast, and then a chondrocyte. I think it's quite good. But cartilage grows either just below the perichondrium. Yeah, so here we have a perichondrium right here. I know this is a bad picture, but it's not that bad. We can have a look at it. And we differentiate two processes in which the cartilage can grow. And the first process is apposition. And this apposition is a process of apposition from the perichondrium. So we have some precursor cells that are under perichondrium here, and they can form chondroblasts and chondrocytes, and they're always dividing here. These blasts are dividing here, and that's a process of apposition, simply. Yeah. So it's really, in a very simple way, we can say it's like the division of chondroblasts and the formation of precursors. Yeah, apposition. But cartilage also is capable of interstitial growth, and that's here, interstitial growth right here, that's happening in the center of cartilage. And this interstitial growth is associated with the formation of groups. So it's really just formation of groups. You know, chondrocytes are able to divide, and when they do so, they create isogenous groups, and that's really interstitial growth. That's what's happening in the cartilage. And one last thing I want to tell you is that when a cartilage does not have a perichondrium, what's happening? Because really not we can find cartilage that doesn't contain perichondrium, and you probably know that joints, hyaline cartilage in joints doesn't have perichondrium. Yeah, because remember, perichondrium is vascular structure, vascularized. Yeah, there are vessels there and also nerves. So it will probably be very painful and uncomfortable to walk with perichondrium in the knees or in the joints in general. So we don't have perichondrium in the like about the cartilage in the joint. So it's growing just by interstitial growth. Yeah, so when there is no perichondrium, there is only interstitial growth, just mitotic division of chondrocytes in the center of a cartilage. Yeah? And it's also this cartilage in a joint that doesn't contain perichondrium is nourished by a fluid that is found in a joint. You know that joint contains fluid like synovial fluid. So the cartilages in the joints are just nourished by diffusion from synovial fluid there. But you probably know that. So just remember, like, walking with perichondrium in the joints will hurt as hell. So that's why we don't have it, yeah, because cartilages are rubbing by themselves and you will lose this perichondrium very quickly. So that's why. So we took over the cartilage and we can go for the bone now. That is even more difficult than cartilage. So let's start again with the main components here. The resident cells or fixed cells, which we have here, are osteoblasts and osteocytes. And again, two forms of one cell. Yeah, osteocyte as cell that is less active. Yeah, osteoblast as a precursor of a cell that is more active, probably more active in terms of production and maintenance of everything and division and stuff. But it's different in the bone because really, once again, the fibroblasts and fibrocyte are different. Fibrocytes were not able to divide. Fibroblasts were those guys that were able to divide in a proper connective tissue. Then we had cartilage with chondroblasts and chondrocytes, and both of them are able to divide. Yeah, so chondrocytes are able to divide because they're forming these isogenous groups. And then we have osteoblasts and osteocytes, and neither osteoblasts nor osteocytes divide. So no division for these guys here. Yeah. So where do they come from, really? They come from precursors that resemble fibroblasts. Yeah, some precursors that resemble fibroblasts from mesenchymal cells. These are called pre-osteoblasts. I'm going to write it down, it's hard. So precursors are called pre-osteoblasts. Or osteoblast progenitors or osteoprogenitor cells. You can find many names for one thing actually, and these can divide. So these two are not able to divide. Yeah, so no division here. Once they differentiate into osteoblasts, they no longer divide. Just remember that. Migratory cells, on the other hand, are osteoclasts that differentiate from these hematopoietic stem cells from monocytes. So monocytes are precursors. Remember this is very important. Monocytes are precursors of osteoclasts, and you can find osteoclasts in bone normally. Yeah, their task is resorption. Yeah, resorption. Because bone, on the other hand, yeah, is very active. It's not as cartilage that was formed and now it stays there and it's not very active, but bone is always doing something, is remodeling, is destroyed and then it's formed once again. We will show you that. So you need osteoclasts in the bone. They're always doing some job there. Yeah. And osteoblasts and cytes, once again, they produce intracellular mass, don't forget that. And the role of osteoclasts is opposite. So they do the opposite. Yeah? So they eat the bone. These guys form the bone and osteoclasts eat bone. But they are not bad guys. Remember, they're not bad. Yeah? You need them. Don't forget, yeah? So you need both of these. So, and then we have fibrillar component of extracellular matrix, and yeah, the collagen fibers. Yeah, so type 1 collagen is important, type 1. She write it like this, type 1. That's forming fibers. It's very important. And also amorphous component is very hard and firm, also like in a cartilage. And the proteoglycans that we have these proteoglycans that contain chondroitin sulfate and keratin sulfate. It's the same as in a cartilage. And here we have a structural glycoproteins as osteonectin. You should remember this, osteonectin, that is connecting everything together. Or we have this sialoprotein osteocalcin. You're going to hear about them more in clinical practice and stuff, so remember these. And what do I want to say? So we have several types of structural proteins here, and this sialoprotein osteocalcin is important for calcium binding mineralization. Yeah, so they bind calcium in the process of mineralization. Yeah, so extracellular matrix contains hydroxyapatite crystals. It's mineralized. You probably know that bone is very hard and firm and stuff. So hydrated calcium phosphate here is a basic mineral in the bone, and these crystals of hydroxyapatite crystals bind to the collagen fibers. Yeah, they bind to the collagen fibers to form a mineralized intercellular mass, really. So yeah, they bind here to these collagen fibers. So if we want to observe bone, if we want to produce these preparations of bone tissue, we have to get rid of mineral component, because otherwise we wouldn't cut it. It's very hard, it's so hard, you're not able really to cut the bone in order not to lose everything or not to lose cells. If you want to see cells that are there, and if you want to see the structure, some kind of, and if you want to stain the slide, you need to decalcify it before. And we use these calcium binding acids for it, or chelating compounds, some chelating compounds. There is also one process that we can use to not decalcify bone, to see it. We can just cut it, really, without demineralization. But these cuts, just simply cuts, are great for studying the mineral component, but the organic components of the mass are destroyed and there are no cells once again. So for our slides in our department, you're able to see only demineralized slides. Yeah, so slides that were prepared in the process of demineralization, and after demineralization then was the cutting, and then you would be able to see the bone. Yeah, so I'm going to show you the picture. So now let's have a look at how the bone tissue really looks like. There is a periosteum. Yeah, we have perichondrium, but we also have periosteum on the surface. Once again, it's made of dense, irregular connective tissue. So there is this dense, irregular connective tissue right here. And you cannot see that very good, but I will show you periosteum once again. And if there is a muscle attachment, we see also these collagen fibers, yeah, collagen fibers, which sink into the bone right here. Yeah, they are attached here to the bone, these collagen fibers. It's really a tendon that is attached to the bone, and they are arranged in parallel, and they are called Sharpey's fibers. So Sharpey's fibers. tissue base fibers. Yeah. So here we can see them. Periosteum is on the surface of the bone, and we have also endosteum right here that is made of loose connective tissue, and that is inside, covering the inside of the bone right here. Yeah, as you can see, bone is hollow, very hollow, and endosteum is covering it on the inside. So we have periosteum made of loose, dense connective tissue, dense irregular connective tissue, and endosteum made of loose connective tissue. If the tendon is clamped to the bone, we see more tissue in the periosteum, as you saw before, this fibrous, and it forms something called enthesis. Enthesis. It's really attachment of tendon and the bone surface. It's fibrocartilaginous right here, so we can see the tendon, the dense regular connective tissue, really, that is really what we can see here, that there is not just this tendon and then we have a bone, but there is a fibrocartilage here. Yeah, the cartilage that contains also collagen type 1. So there is a space made of fibrocartilage. And after that, here we can see the mineralized cartilage. And after mineralized cartilage, we have a bone. Yeah. So there is not just periosteum and collagen fibers of the tendon, but there is this enthesis that is firm. Yeah. So we have the tendon, the fibrocartilage, mineralized cartilage, and after that, the bone. This is forming this insertion site right here. Yeah, you probably wouldn't see that on our slides. It's just, well, I should tell you that it's not, there are some other connective tissues between, yeah, the bone and the tendon, the fibrocartilage, the mineralized cartilage also. So, what can we see here? Well, when we look inside the bone tissue, this bone tissue is often formed into such trabecules. Yeah, into trabecules. So this is a picture. This is a trabecule right here. Trabecule. We see that there are other cells on the surface of the trabecules, and other cells are on the inside. Yeah, inside in the trabecule. So here we have some cells on the periphery of the trabecule. On the surface, here we can see these guys here. On the surface, we can find osteoblasts. Here, osteoblasts. Remember, these are precursors of osteocytes, but they are not able to divide. Yeah? So one osteoblast is forming one osteocyte in a process of just differentiation, because these osteoblasts, what are they doing? They're producing mass, they're producing bone matrix, and when they do so, and they lose themselves, and when they are embedded in this trabecule, in the matrix, they differentiate into osteocyte. Yes, so they're not dividing, they're just differentiating in the less active cell. Really, these osteoblasts, they kind of look like epithelium, but remember, these are not epithelial cells. Yeah, they're organized into epithelioid fashion. This is just epithelioid fashion on the surface of trabecules, but these are not epithelial cells. What does it mean, the epithelioid? It means that they resemble epithelium, but they're not epithelium, because they don't contain cytokeratin filaments inside them, and they also don't sit on the basal lamina. Yeah, so if this was epithelium, there should be a basal lamina on the basal surface, but it's not there. And these are not epithelial cells, they just look like epithelial cells. So there's no basement membrane, and they also don't contain any specializations on the lateral surfaces or in the apical surfaces, because they're not epithelium. Osteoblasts are sometimes cuboid, cubic, like this one here, and sometimes are more columnar or cylindrical, like these, and sometimes here we can see place where they are flat. So there are many shapes of osteoblasts, really, and it really depends on their activity. When they are more active, when they are producing, we can see it here like these dots, they're producing, they're bigger because they need space for the organelles. So they need some organelles in order to produce stuff, and they need also some space, so they're bigger when they're producing. The more active osteoblast, the bigger osteoblast. And they're producing a bone, of course. And the newly produced or newly formed mass is called osteoid. So it's not mineralized. Osteoid, it's not mineralized, and it's this really matrix just below osteoblasts right here. So newly formed bone right here. It's not yet mineralized, as I said. It consists only of an organic component like collagen, glycosaminoglycans, glycoproteins, and proteoglycans. When it begins to be mineralized, we call it the bone matrix by default. So mineralized is bone matrix, really, and osteoid is non-mineralized matrix. And you can see osteoid sometimes also in the bone. So you should be able to describe this layer, this light eosinophilic layer below osteoblast, newly formed bone there. As matter gradually increases, it sometimes happens that some osteoblasts, like for example this guy here, some of them remains embedded in the mass. They are like, okay, I've produced this, so I want to stay there because I've done this. I want to be here in this matter. So they set aside, and they form osteocyte. They differentiate into this osteocyte. So these things, it's written here, these cells right here are osteocytes. They also are in this space, lacune. We also call it lacune also in a bone. So we have lacunes in a cartilage and we have lacunes in a bone. So there are lacunes everywhere in the cartilage and bone. So these spaces are lacunes right here. But they're alone there. Don't forget, alone. There are no isogenous groups here because osteocytes are not able to divide. Remember that. These are not dividing. Osteoblasts and osteocytes. No division. Among osteoblasts, we sometimes can observe osteoclasts here, these big, huge cells. Osteoclast. This is one big osteoclast right here. And they are very huge. They are the big guys there. And they have many nuclei, as you can see it here. Many nuclei. And why is that so? It's because they are formed by the fusion of its precursors. So there are many monocytes that were here before from the cells, from the bone marrow. They come here in this tissue and they fuse together and they form osteoclast. Osteoclast. So it's a kind of syncytium. It's like syncytium. Many cells fuse together and they form this osteoclast right here. So it's very huge. And sometimes it's written in some textbooks that you can sometimes see 50 nuclei in one cell, but I've never seen such osteoclasts really. So there are probably just like in here 10, 20 maybe, yeah, nuclei. Here we can see 5, but this is just a small osteoclast. Sometimes you can see 10 nuclei in osteoclast. It's a very, very, very strange cell, I think. And really, it's like 50 nuclei, that's enough, even for me. So, osteoclasts really are used for bone resorption. Yeah, so these osteoblasts are making the bone, and osteoclasts are resorbing the bone, on the other hand. And we should probably look at some picture. Yeah, here, for example. Here we can see a trabecule, a bone trabecule right here. This is a trabecule in some kind of tissue, probably new forming trabecule right here, because we can see some tissue around. So we're probably forming bone. And we can see these cells inside, alone in their lacunes. These are osteocytes. And these osteoblasts on the surface of a trabecule from one side and also from another side. Yeah, in epithelioid fashion. So there is some kind of organization of osteoblasts, and we can see these big cells right here, and these are multinucleated osteoclasts. We can see 3 nuclei right here in this cell, and also here we have osteoclast right here. So they lie on the surface of the bone, and really these osteoclasts do not enter the bone matrix. They're always on the surface. So for the osteoblast, they can really enter, and when they do so, they differentiate into osteocyte. But osteoclasts, they just sit there and resorb the bone, so they don't go deeper, these things. Well, probably because they're so big, they can fit into the bone, yeah? So they're just too big to enter. Yeah, and these are some pictures of osteoblasts. We can see there is brighter or lighter nucleus in the osteoblast because this osteoblast is more active. There are some protrusions going outside, yeah, we can see it right here. And osteocytes have darker nucleus right here. They are embedded in the intercellular mass. And these osteocytes also have protrusions, but they are very long and very thin. Yeah, they're very long and very thin, and they are branching also. Yeah, and they are connecting with protrusions of another osteocyte. This is very important and crucial for the bone, and these are called philopodia. So very, very long protrusions, very thin and branched philopodia. These protrusions are located in the canals of the bone called the canaliculi ostium. So they are in the bone in canaliculi ostium. We can see them also. These canaliculi can be seen in a light microscopy. We are really not able to see these protrusions. You're not able to see philopodia because they're so thin, but you can see these channels, these canaliculi in the bone. I'm going to show you so you believe me. They're like spider-like, really. They look like little spiders right here. They're a little bit scary, but since it's Halloween, or it was Halloween, oh, doesn't matter, but they're really located in these channels, and they are connected. They are connecting two and more osteocytes because they are transferring substances, yeah, between osteocytes. This is crucial, this is very crucial for them, because otherwise they would not be able to survive. They would not receive nutrition. Yeah, so they're transferring food, really, pizza and stuff. Yes, I'm hungry right now. So they really inject nutrients through their protrusions, and these filopodia create entire network. Yeah, they connect with these connections that you probably know already. These are nexus. So there are nexi, or nexus is one communicating channels between these protrusions. Yeah, so here we can find many of them here. Yeah, so we can see it right here. There is a periosteum on the surface, dense irregular connective tissue. This is a bone, highly eosinophilic structure. We can see osteocytes in the lacunes right here. Osteocytes right here. And here we can see a layer of osteoblasts right here in epithelioid fashion. We can see these big channels in the bone. I'm going to explain later. Yeah. And these channels are filled with loose connective tissue with some vessels, and there are also some cells, probably some immune cells are here. Yeah, so we have normal vessels. These are erythrocytes right here. But for what's important for us right now, these are osteoblasts. Yeah, organized here. These are osteocytes. And I don't know if you can see it, but I can see that there's like this spider-like things going outside of these cells, and these are the canaliculi that are containing filopodia. But sometimes you can see it even more. Yeah. So we can see really ground section of a bone. It's non-demineralized bone, opposite, non-demineralized. So we can see the structure very well, and we can see these canaliculi here once again. And everything is connecting together. So this osteocyte right here is connected to this one right here, and this one is somehow connected to this one with the nexus right here, the connections between these protrusions, and they are transferring substances. Yeah, substances. So probably here it's not here right now, and we're not able to see osteoblasts here because they're gone due to this process of preparation. Yeah, and there should be also tissue with the vessels here, but they're not here. But imagine here is a red blood cell that nourishes these osteocytes right here, and these guys are very good nourished, and they transfer nutrients to this one right here. Yeah, so that's how it's working. So you should know about these filopodia right here, processes, because they're crucial in the nourishment of the bone. Yeah, we're going to learn more about this structure. Yeah, so osteocytes are cells that maintain the matrix. So we know the osteoblasts are producing this matrix, and osteocytes can also produce, but they're more for the just management or maintaining the matrix. They have the ability also to produce, but this ability is muted, so they are at rest, I guess, maybe. However, they can be stimulated. Yeah, they can be stimulated so that osteocytes are found in this formative state. Yeah, this is the quiescent state. It's in rest. Osteocyte in rest. When it's stimulated, it's called the formative state of osteocyte, and sometimes they can even resorb the bone like osteoclasts. They can resemble them. They can resemble osteoclasts, but and then they are in the resorptive phase. Yeah. So they can be specialized for resorption, also these osteocytes, but this ability is more prominent in osteoclasts. Okay. In this photo, we can see osteoclasts right here. So here is nice osteoclast here, and maybe the third one just a little bit smaller, or it's somehow sectioned not so good than these two. But we can see multi-nucleated cells here, and the cytoplasm of these cells is eosinophilic. Like macrophages, they also have eosinophilic cytoplasm because they contain a lot of mitochondria and stuff. And let's have a look probably on some osteoclast. There is also a huge guy right here, so don't forget it's formed by fusion of many monocytes from the blood. And here we have a nice osteoclast right here. So again, there are multinucleated cells. The cytoplasm is very eosinophilic. They contain a lot of mitochondria and lysosomes. Yeah. And where they touch the bone matrix, here they sit on the bone matrix, they form some kind of depression there. So they're huge, they need some kind of space where to sit. So they form this depression, and this depression here, when they sit, is called half-shape lacune. Half-shape lacune. Just a depression in the bone matrix. Yeah? And it's not enclosed space. Like a lacune, normal lacune. It's just like a half of lacune, or I don't know how to explain, but it's just a depression. So they're not covered in something. At the point where the osteoclast is adjacent to the lacuna, or the lacune, wavy rim is formed. This wavy rim is formed. And osteoclasts secrete lysosomal enzymes outside in the area of rim. So really this osteoclast that is sitting right here secretes lysosomal enzymes here. And probably you should now think about like, wait, what? That's strange. You're secreting organelles outside? Because lysosomal system, lysosomes are organelles, yeah? And you secrete organelles. Normally cells don't secrete lysosomes outside. Yeah, they are enzymes that are in the cell. They are important in the process of digestion, but normally cells keep their lysosomes inside. But osteoclasts are different. They secrete them outside, and they are crucial for bone resorption. Yeah, but you know that lysosomes need acidic environment. Yeah, the lysosomal transport vesicles need to fuse with endosomes in order to function, in order to activate enzymes. So lysosomes need acidic environment. So osteoclasts also need to provide this acidic component, yeah, for the lysosome enzymes to function. And it does so through a chemical reaction right here. And this chemical reaction is catalyzed by carbonic anhydrase. I'm going to write it down. Now you think it's like, why am I learning about this? But it's crucial in pharmacology, because sometimes you block this carbonic anhydrase in order to osteoclast to stop functioning. So you should learn about it. Anhydrase, carbonic anhydrase. I hope I wrote it correctly. So it's a catalyzer for this reaction to happen, and you see you form these protons, and they are pumped outside to this place, and we created acidic environment here with low pH. So now enzymes can work, and they are activated here. Yeah, so this is a very important reaction, and really you need a lot of energy to do this. This reaction needs a lot of energy. This proton pumping needs a lot of energy. so you need energy a lot. So you need a lot of mitochondria in the cell, and that's why the cell is eosinophilic. Yeah, so eosinophilia, high eosinophilia. Osteoclasts are able to penetrate the bone and form such channels or passages. Really, they form passages, and this gives the bone remodeling ability. Yeah, the bone can remodel, and it's very important for bone to remodel and be produced once again and do stuff and be active. Yeah, so these osteoclasts aren't bad guys; they're good guys. And really, there are some types of bone tissue, and we differentiate primary or woven bone and lamellar or secondary bone. You should know this division, it's very important. And when the bone tissue is formed in embryo, the fetus, or in healing, yeah, you broke the bone and now you want to heal the bone, so you need bone formation in healing. This woven, the primary bone, is made. Yeah, so first, it's always like that. First you have woven bone, primary bone, that is differentiating into lamellar secondary bone. Yeah, so once again, it's temporary bone during development and repair. Yeah, that's for us important. It has irregular arrangement. Yeah, irregular arrangement and low mineralization. Yeah, then it differentiates into this lamellar secondary bone, which is built of lamellae of parallel collagen fibers. So lamellae are just these parallel collagen fibers that are very thick, and these collagen fibers are embedded in mineralized matrix. You can find osteocytes mostly in between these lamellae within cementing substance. So there's some kind of cement in between, which is connecting these lamellae together. So they are in the cementing substance. And we divide this lamellar bone, the secondary bone, into compact bone, yeah, which is made of osteans or Haversian systems, or spongy bone, which is like made of mostly, as we saw before, these trabecules. Yeah, so anastomosing or connecting trabecules together, and maybe there are some parallel lamellae or osteons there. So, but they're just like these trabecules. It's not so compact. Yeah, there is much more space in between these trabecules than in compact bone. So, and we're going to have a look on them. Yeah. Once again, we have this primary bone, woven bone. Yeah. So what can we see here? There is some connective tissue. This is very primary bone, so we have just some like these islets of bone right here. Yeah, very eosinophilic. Yeah, they're very eosinophilic. They're made of these trabecules of bone. Right here, what we can see, these are osteocytes in the middle. On the surface, there are osteoblasts. Yeah, on the surface. And here we can see some connective tissue around, so probably this bone is just forming, yeah, itself. And really, it's permanent. This type of bone is permanent only in the cranial seams, so these connections between bones of the cranium. So you can find this primary bone here, but it's usually only a temporary tissue. It's not mineralized that much. These fibers are really disorganized. These collagen fibers here are disorganized, so we're not able to see some kind of organization. You will be able to compare that to another picture. So no organization here, just some eosinophilic matrix made of collagen, with osteocytes inside and osteoblasts on the surface. So There's no organization. And this bone is transformed into lamellar, the secondary bone. And really the secondary bone in humans predominates, really. On the other hand, a lamellar bone, the secondary bone, has a regular arrangement. There should be periosteum on the surface right here, but it's not. It's gone due to preparation somehow. It should be here. You should have periosteum on the surface of your bones, but okay, never mind, it's not here. The lamellar bone really is forming these lamellae, which are regularly arranged, and we can see them really here as a sheath lamellae right here, parallelly arranged. Lamellae are just collagen type 1 with these amorphous components together. So it's just everything I said before, the extracellular matrix that is together. It's forming these lamellae. It's also mineralized. But this is a slide that was created from demineralization. So we can see the high eosinophilia of the collagen, yeah. And these are sheath lamellae, and sometimes they are, they are concentrically arranged. Also these canals here, yeah, these canals. And lamellar secondary bone is forming something called osteons. So this is, for example, osteon or a Haversian system. We're going to have a look on it. It's just like some kind of canal in the center, and around we can see these concentrically arranged lamellae, and they are together creating osteons right here. Yeah, and we can see lamellae also here. And remember, there are lamellae. So this is one lamellae, this is the second one. And between them we can see this lighter substance here, and this is cementing substance right here in between the lamellae that is connecting them together, cementing substance, just, yeah, right here. And we can see also nice osteocytes right here, and normally you should be able to see osteoblasts that are on the surface of these channels right here, but they are also not there. It's sad, yeah, but this is picture for you to see the lamellae right here. They are also sheath lamellae on the surface of the bone, like here, and there are also concentrically arranged lamellae around the channels, forming this Haversian system that we can see here. That is very, very cool picture from scanning microscope. I really like it because really we can see this canal here in the center going nowhere to some other space, or I don't know, but it's scary kind of. And we can see many concentrically arranged lamellae here. So this whole structure, I can show you, this thing right here is this osteon, or in other name Haversian system. And we can see these lacunes that don't contain osteocytes because of the preparation, yeah, they were destroyed. And they are in this cementing substance between lamellae, so they are located in this cementing substance. And this picture is also in oral exam, so have a look on it, yeah. So very important picture from scanning microscope. So we can see only surfaces, yeah, but this was like the gutted bone. It's not demineralized, it was just like gutted, and that's why we are not able to see cells real here, only this empty channel here that normally contains loose connective tissue, and this channel also normally contains osteoblasts that are sitting here. So there should be osteoblasts sitting here forming the bone, but they're not. And osteocytes in the lacunes here in the bone matrix. Yeah. So when the lamellae are so concentric, we call this whole system osteon or Havers system. Haversian system, and we can see it right here in the bone. So this is an example of compact bone, very dense bone, made of these Haversian systems right here. Yeah, so many concentrically arranged lamellae in the around canal that is also called Haversian canal here. But how is this canal formed? It's normally forming probably right now in your bones, this canal. So what's the cell that is eating bone, that is resorbing bone? Well, osteoclast. Osteoclast, this huge guy. So in the bone, huge guys in the bone that are formed in the process of fusion of many monocytes, remember? Yeah, resorbing bone. So yeah, that's really what's happening in the bone. How do we form osteon? How do we form this canal and arranged lamellae around? Is that, yeah, these are osteoclasts, they're eating the bone, they're forming this very good channel right here. So they form the tunnel, and here they are at the bottom of the tunnel. They were originally on the surface in hot-shaped lacunes, but they broke into the bone tissue where they formed a channel with a relatively wide luminous. Yes, so at the beginning of the formation of osteon, we have very wide Haversian canal there. So more wider the Haversian canal, the newer the osteon there. Yeah, so newer osteons contain wider Haversian canals, because what's happening? Okay, so osteoclasts are resorbing the bone, they are forming this channel. Normally they are sitting in their hot-shaped lacunes, but sometimes they decide, okay, I'm going in. Yeah, I'm going in and I'm resorbing the bone right now, and I'm creating this huge wide channel. Yeah, not only one osteoclast, but many of them decide to form it in the process of signalization, and because of the pressure to the bone, and because of walking, running and stuff that is happening with your bones when you do something, not just sitting like right here. So yeah, they form the channel, and what's happening here, osteoblasts start to migrate here, and they start to produce bone mass, and they start to produce lamellae here. Yeah, so there are many generations of osteoblasts, and they line the canal and began to form intercellular mass. So they form lamellae gradually. It's not like all lamellae are made at the same time. They gradually form these lamellae one by one. So these brown lamella that was here, the first one, that was the first one that was made actually. The first lamella here was made, and then the second one was made, this pinky one here. Yeah, also for the other side here. So this brown was the first one, and the pink was the second one. So, and there are more and more generations of osteoblasts there. Yeah, some of them force, I think they die. The other osteoblasts come to the place, they form another lamellae, they die. So it's more like some kind of family business, I guess. Not so perspective, but somebody has to do it. So yeah. So the further from the center, the older are the lamellae, really. So they are narrowing this channel in the center. They are narrowing this osteon in the center, this canal, and this canal will close gradually. Yeah, so sometimes the osteons close, there is no channel. Yeah, so we will see that on other pictures here, how they are formed. Yeah, they are some kind of first generations Haversian systems, then there are second ones. Yeah, they gradually close these first generation systems. Yeah, so more and more lamellae are formed. Sometimes you're not able to see them. Yeah, so you're able to see only the newly formed Haversian systems in the bones, because they are going above each other. Yeah, So, and sometimes because of that, you can see only those parallelly arranged lamellae somewhere here, and you're not able to see the whole system there. So there are many, many generations right here. And that is really how the compact bone looks like. Oops. I'm sorry. So this is like the three-dimensional diagram, and of course the fibers can also be seen here. Yeah, we can see these collagen fibers here, and what we can see there is that in adjacent lamellae they're going in the opposite direction. Yeah, so these collagen fibers are going this way and these in this way. They're almost perpendicular, yeah, to each other, and this ensures the strength. Yeah, that's why the bone is so strong. It's because of this arrangement. They're going perpendicular, they're crossing each other. Yeah, so they're forming a system like this. And we can see in this substance between lamellae there are these osteocytes with these processes, filopodia, connecting to each other, and we can see these canals that were formed with osteoclast, Haversian canals. Yeah, they are in the axis, they run with the bone axis, these Haversian canals, but we also have these another canals here, and these are Volkmann's canals that are perpendicular to the axis, and they are connecting these Haversian canals together. There are no concentrically arranged lamellae around Volkmann's canals. Yeah, so there are lamellae, concentrical lamellae organized around these Haversian canals, but they are not organized here in the Volkmann's canals. I'm going to show you some pictures. Just remember right now, in order to really nourish the bone, you need this system, because Haversian canals and also Volkmann's canals contains loose connective tissue with vessels. Yeah, so we need to connect everything together, and then it's nourished by the process of these processes, these filopodia processes in osteocytes. So it's really not only like diffusion, they're actively transporting substances. So the situation is different than in cartilage. It's very different. And that's why you can form also new bone, and its bone is also always remodeling and is active structure. Yeah, cartilage, okay, no, just diffusion, no system really, just maybe some fibers, and sometimes even no fibers in hyaline cartilage, because type 2 collagen doesn't form fibers and stuff. This is completely different. Yeah, if some of you tell me like the cartilage, oh, almost the same as bone, it's hard and firm and stiff and nothing else, like, no, gosh, no, it's totally different structure, and you can see it right here, the organization, the nourishment and everything. The cells, they are not able to divide, but still you can produce new bone, really, because of these precursors, pro-osteoblasts, that are under periosteum. Here we have a picture from light microscope. Here we have Haversian canal right here. It's more longitudinal section, but we can see these are lamellae that are around, yeah, arranged. Here we can see them. There is a connection, Volkmann's canal, canal. And here we have another Haversian canal here. Yeah, no, so we can see these lamellae. Probably cross-section will be better, but this is a very rare picture. We can see it quite nicely. Some canal that is connecting these two Haversian canals must be Volkmann's canal. Yeah, this is really... we can see that these spaces between osteons gradually change, because we have more and more osteons forming right here, and these older osteons, the first generation, the second generation, form lamellae that are called interstitial lamellae. So There were normal lamellae of an osteon before, but now when the new osteons are formed, they're called interstitial lamellae, right here. We see two photos here. On the left, this is the calcified bone right here, so it's normal hematoxylin eosin here, and here we have just cut through the bone. Yeah? So this bone wasn't decalcified. Yeah, this was decalcified, this wasn't decalcified. These are just comparison of two pictures. And here we have a Haversian canal right here, and here we have another Haversian canal right here, and this right here is wider, it's wider than this one. So probably this osteon right here is newer, is brand new, not brand new, but it's younger than this old one here. Yeah, the younger, the wider the canal. So that was a compact bone, and now for the spongy bone right here. And spongy bone is made of these trabecules, but also you can find osteons and Haversian system also in these trabecules. Yeah, so it's not irregularly arranged like in woven bone, but you have these trabecules also like in woven bone, but you can find osteons there and Haversian system there. You can see right in this picture there is a bone marrow between these trabecules. Yeah, right here. So we can see it right here, and this is the bone. These are the trabecules of the bone right here. This is how it looks like, yeah, in the photo. It's very, very, very nice photo, and this is just a system of a spongy bone right here. This is also how it looks like. This is bone marrow made mostly of adipose tissue, so it's a bone marrow in adults, yeah, mostly adipose tissue right here in between. And these are the trabecules right here, these trabecules. So it's kind of some kind of different picture from different staining, but it doesn't matter that much. So, and for the last parts here, how do we form bone? Yeah, how do we form bone? Really, bone can be formed just like cartilage in the basis of mesenchymal tissue. It's called intermembranous ossification or desmogenic ossification, or on the basis of cartilage. But now let's have a look at this. So intermembranous ossification is bone formation on the basis of mesenchymal tissue. So first, the mesenchymal cells are just connected together. They form this bone blastema, just like we had cartilage blastema, we have also bone blastema. So this group of mesenchymal cells connecting together, and they form these osteoblasts, which are then forming or producing a bone matrix. Yeah, so it's really just straightforward, like in a cartilage formation. So star-shaped mesenchymal cells, then we have blastema, yeah, that form, there's one step in between pre-osteoblast, yeah, so first we have pre-osteoblast, then we have osteoblast, then they form this primary bone tissue, yeah, primary bone tissue, so no osteons, no arrangement or anything else. And these osteoblasts, then some of them turn into osteocytes that are embedded in the tissue right here. Yeah, these osteoblasts arrange themselves in this epithelioid rim on the surface. Yeah, and we can see it in this picture right here. This is an intermembranous ossification. We have also a slide about this intermembranous ossification. And when these little islets of the bones are formed, these little islets are called spicules. Yes, spicules, these. And they are fusing together, and not like moving, but they're connecting in the process of bone formation from mesenchyme, and these create bigger structures, and these are the trabecules right here. Trabecules. Yeah, so spicules fuse and they create trabecules. And we have osteoblasts on the surface right here, and they are still forming the bone, the new bone. And this is the desmogenic or intermembranous ossification. This type of ossification creates a skull, a whole calf, and some facial part, yeah, of the skull, and also a clavicle is created here, in the process of intramembranous ossification, clavicle, and but also long bones grow in the width with this desmogenic ossification. So they grow wider and wider with desmogenic ossification also. Yeah. But really it's happening, this desmogenic ossification is also happening in the beginning of chondrogenic ossification. So remember, skull, calf, yeah, the whole calf, and some facial parts, and the width of the bones, and also in the beginning of process of chondrogenic ossification. Yeah. So probably this around here in this desmogenic ossification here, this tissue around, it's not actually mesenchyme. This not really doesn't look mesenchyme like this. It's more of a loose connective tissue. Yeah, it's more differentiated than mesenchyme, but it still is not so differentiated tissue here. So it's just some kind of mixture, yeah, around here. Okay, and the chondral ossification, well, this is much more difficult. This is much more difficult because it's difficult. And you form long bones with this endochondral ossification. And really at the beginning of chondrogenic ossification, yeah, I'm going to call it chondrogenic ossification, we have the mesenchyme. First, we have mesenchyme once again. Oh, there's always some mesenchyme on the beginning of everything. Mesenchyme. So we have these mesenchymal cells, and first it forms, this mesenchyme forms cartilage. So we need some model. Cartilage. We need some model, and based on this model we can create bone. So first we form cartilage from the mesenchyme, and this cartilage is hyaline. So don't forget, we have a hyaline cartilage forming in the process of bone formation first. So this was the second step. And we know that hyaline cartilage is avascular. So it doesn't contain vessels. It's very hard to nourish the cartilage. It's dependent on diffusion. And in this case, yeah, desmogenic ossification begins in the perichondrium. So the cartilage has perichondrium on its surface, and this perichondrium, this connective tissue, dense irregular connective tissue, starts to ossificate. Yeah, it starts to transfer itself into bone in the process of desmogenic or intermembranous ossification. That was the first ossification that we described here earlier, just a few minutes earlier. Desmogenic ossification. Yeah, so we form really a bone collar in the perichondrium. of this cartilage. And really what's happening, this bone color, is created around the diaphysis, in the perichondrion. It's really a thin layer of bone tissue that's created around the diaphysis of hyaline cartilage. And the diaphysis, the cells in the diaphysis of this still cartilage will respond to that, because now they're like, what the hell, we don't have enough substances, we don't have enough nourishment, because really there is some kind of bone collar that is choking the cartilage and there is no diffusion going on of substances. So, and the cells in this cartilage start to respond to this undernourishment or malnutrition in a way that they start to proliferate. So it really doesn't make sense at all. It's like, let's undergo mitosis, shall we, when we don't have enough nourishment here? Like, when I'm hungry, I don't want to divide. I'm just hungry. I want to eat. So, but cells like chondrocytes are different. So they start to proliferate. They feel like there's some kind of need for them. Like, maybe more of us can survive somehow. So, but really they exhaust their mitotic activity. So really the chondrocytes, I'm going to write it down for you. Chondrocytes, first, what are they going to do in this response? This is very bad for them, this bone color. First, proliferate. Once again, for me it doesn't make sense, but whatever, do what you want. It's okay. But they just exhausted right now because they divided, and they began to hypertrophy. Next, they began to hypertrophy. And why? Just think about it. They try to store everything that is around. They're like, oh hell, we're going to die. So every chondrocyte is just storing everything. They hypertrophy, that means they're bigger and bigger, storing substances, storing lipid droplets and glycogen and stuff. And they like resorbing surrounding mass. But also they hypertrophy, but also they starting to produce enzymes. Next. They start to produce enzymes because they're like, oh my gosh, I need to do something right now. I'm dying. So enzymes outside of me. But what they really do, like, these guys are crazy, but what they really do, they produce alkaline phosphatase. And these guys, alkaline phosphatase is important in the process of calcification. So what's happening? Calcification. And well, this is the end. Because when you calcify everything around you, it's not going to help in the nourishment. You're with me. It's not going to help with nourishment. So when you calcify everything around, it's just like they're dead in this place. So they're going to die. I think they could handle it better, but it's not happening, so they die, really. So first they proliferate, then they hypertrophy, they produce enzymes, the calcification begins in surrounding tissue there and in surrounding cartilage, and then, well, they die because there is not enough substances here. But it's not their fault, but actually, like, this system is not very good. But the bone can form in this case because really there will be empty spaces that were filled with chondrocytes before, and these empty places that are going to stay there as Dr. Uhlich said in his presentation, that nature does not like empty places anywhere, and it fills them with tissue and blood vessels. So when something died in some place, it's replaced with something. So the cartilage, the cells in the cartilage die, yeah, and they are replaced with tissue and blood vessels, and that's where the like the chondroclasts, yeah, are able to transform here, and these chondroclasts will eat the rest of the cartilage in here, yeah. So yeah, and after that the precursors can come here. The precursors of osteocytes, osteoblasts, can come here and form the new bone, yeah. So this is very difficult process, and once again we're going to cover it. I'm going to show you some pictures and once again explain how it's happening right here, yeah, what's happening. At the end we form these epiphyseal plates right here when it's still hyaline cartilage, and it's slowly transforming into the bone so you can grow, yeah. Yes, and here we have nice picture from embryo. Here we have something called primary ossification center right here, primary ossification center. It's forming in a diaphysis. So first you form a bone in the diaphysis, and after that you form a bone in the epiphysis of the bone. So this is, okay, so this is one bone right here. Yeah, so this is a foot right here, and this is one bone in a foot. And here is epiphysis, and this is epiphysis. I should label it with E. And here we have a diaphysis, and the first primary ossification center forms in the diaphysis, and the secondary ossification centers form in the epiphysis. So one is here and second one is here, but we are not able to see them. We're only in the beginning of bone formation, and we can see only this primary ossification center in the center, yeah, in the bone. So there is some kind of bone marrow, bone cavity is also formed, yeah, and bone is forming right here. And this process of bone formation, the bone color formation, will also happen in the secondary centers right here, right after, yeah, and creating also the process is the same. Creating the secondary ossification centers, the process is the same. Just the copies, the process copies the primary. And the hyaline cartilage after that will remain only between diaphysis and epiphysis. And also in the articular surfaces, of course, yeah, where we can't find a perichondrium there. So we can see it right here. This is bigger place where we can find bone, like cavity here. Bone marrow will be here, or is here right now. And here we have also hyaline cartilage in one place and hyaline cartilage on the other hand. Yeah. And I made a mistake. Probably these are not toes. These are fingers, fetal fingers. Yeah, right here. So probably just fetal fingers right here. And this is just for another photo, just you can see this epiphyseal plate that is forming bone, but we will look in better detail right now what's happening. This is epiphyseal plate right here. We can see that from the cartilage right here, it's gradually transforming into the bone, which is here. You can see the difference between bone and the cartilage right here. Cartilage is less eosinophilic, this lighter pink. than the bone, and we can see these isogenous groups that are here. We're not able to see that in the bone, so sometimes make a problem for students to recognize bone and recognize cartilage, but don't worry, you will get there. But yeah, this is really gradual bone formation right here. We can see really there are just separated here chondrocytes in the isogenous group, but they are proliferating. We can see many of them here proliferating right here. They hypertrophy right after, so they are bigger and bigger because they want to store substances, and they die, yeah, because of the calcification that is going on. This is a calcification. You can see it's darker, yeah, it's darker than here. So here we can see also calcification right here, yeah, little more like it's basophilic, more basophilic area here, and then they will die. Yeah, so here we no longer can see any chondrocytes. We can see these empty places right here. So probably there are chondrocytes, and they die, and there is just empty space. Yeah, empty space right here. But yeah, pro-osteoblasts migrate here. Yeah, migrate here, and they form osteoblasts, and they form into osteocytes. Yeah, so this is a place for them, happy place. So the tissue penetrate here, yeah, and osteocytes can form here in this place right here. And we will now describe it even in more detail, or I can show you pictures from higher magnification right here. So here we have zone of reserve cartilage or intact cartilage right here. Nice little cartilage here, nothing special happening here, but it's happening somewhere here. They don't have enough food, nourishment, so they start to proliferate, as I said before. It's like, okay, we don't have much food, so let's have sex. Well, it's not sex, but yeah, you know, they're undergoing mitotic cleavages, so they start to proliferate, and we can see these isogenous groups right here that have many people inside. Many people. I'm confused right now. But here we have a zone of hypertrophy, so we can see they're bigger and bigger. They're eating a lot because they want to save themselves. And then we have a zone of hypertrophy and calcification. We can see these little basophilia, because remember, they start to produce alkaline phosphatase, ALP. You can measure LAP in the blood, really. So it's important. And really, this enzyme is mineralizing the bone, so it's very important for bone mineralization. But eventually the chondrocytes will die because of the calcification that is happening around, that worsened the nourishment of the chondrocytes. So they start to die, really. This is called zone of erosion when they are dying. Yeah, this is really the place where they are dying right here. So we can see chondrocytes right here, but we're not able to see them here. And this is this empty lacune that will be filled with precursors of osteocytes right here. Yeah, and these osteoblasts that were formed from pre-osteoblast starts to form osteoid. Yeah, but what's really happening here, what we can see here in this picture, we can see, I'm going to label it with this red, we can see still cartilage here. We can see the cartilage here, the matrix. We're not able to see cells, but we can see the extracellular matrix here. And we can see also darker layers. These layers are more eosinophilic, and this is where osteoid is formed. So these cells right here that are sitting here, these are osteoblasts. We can already see osteoblasts here, yeah? And also here. And these are forming osteoid. So this layer here is osteoid, right here. So this is the zone of osteoid formation, and then this osteoid is going to mineralize, yeah. So then we have a zone of ossification, darker zone here, yeah, darker zone here. So we can see the zone of really ossification right here. There's a bone formation. We can also see osteocytes right here, and this light space inside, this light thing inside here is still the old cartilage that is slowly going to transform into the bone. Yeah, it's going to be transformed into the bone. In that process, that it's not like it's really transforming. Yeah, I said it's going to transform. It's not like it's transforming. It's really these cartilage matrix that stayed there because cells died, yeah, but matrix stayed there. It's resorbed by those cells, as I told before, the chondroclasts, yeah, they're going to eat everything, the old cartilage. So remember this thing, like you need cartilage in order to form bone, but it's not that cartilage is transforming into the bone. It's not that simple. Yeah, you destroy the cartilage and then you form the bone, and you have these cartilage like trabecules just to inform the osteoblasts where they should form the bone. Yeah, there's just like signs for the osteoblast. They're not transforming the cartilage. And then we have a zone of resorption, which is here, because when you really they want to make bone so much, they make many of it, and you still need some kind of bone marrow inside, so you need to resorb also the bone in order to form this bone marrow inside the bone. Yeah, so there is also a zone of resorption when you once again want to resorb the bone, the newly formed bone. Yeah, so that was all. Very difficult process. I know there is much more studying around and stuff, but you should know it like this. As I said it to you right now, you should know this basic principle. Yeah, you should be able to tell that it's formed it's formed the cartilage from mesenchyme, then it's destroyed, and why it is destroyed, and what are the zones called. Yeah, you should know how to describe these zones, and then its new bone is formed. And very last part is going to be about bone repair, then some joints. Yeah, we're going to describe joints. Bone repair is kind of simple. But it's bone like healing, like after the fracture. First, as I told you, first the fibrous bone forms, or not the fibrous, the woven, the primary bone is formed. Yeah, so the periosteum proliferate. Yeah, periosteum proliferate, and it forms first. It forms hyaline cartilage there. It forms hyaline cartilage, but as I said before, you need some kind of model, yeah? And then you form the bone from the hyaline cartilage, yeah? And this process is very long. You should you shouldn't move the bone, fractured bone, because in that case you can make a joint, a pseudo joint here. So it's not very good, and you form something called the callus, yeah. So you have a callus right here that is formed by primary bone, yeah. Then you form the secondary bone, which contains these osteons and lamellae, and then it's healed with secondary bone right here, normal. So bone can be healed very good, yeah, when you're not moving with the fracture. So remember, it's the healing is based on the hyaline cartilage formation, yeah. And you form this primary bone, and then you form this secondary bone from the primary bone. So that's it. And we have some kind of joints. Some joints you can move very easily. They're called diarthroses. These are the joints, normal joints, in the human body, like the knee or in the arm or in the fingers, like normal joints that you can move. But synarthroses, these are just slightly mobile growth. They may be different tissues between some parts of something. I'm really struggling with explanation right now. So when the bones are distinctly ingrown to each other, such as the sacrum, so this is example of sacrum, it's called synostosis, sacrum. So they're just bones that ingrow together the sacrum. These bones can be connected with cartilage. For example, it's called synchondrosis, when there is cartilage inside two bones, or syndesmosis, for example, in cranial sutures in children. Syndesmosis right here, when there is connective tissue in between. And the movable connections are diarthrosis. But these joints are more for the anatomy than in histology, because we don't really have slides to show you, but you should know basics about them. But it's just more anatomical section, I guess. And really, for the joints, they are formed by two epiphyses, or epiphyses and something else, some kind of other part of the bone. They are covered with articular cartilage right here. Most of the time is normal hyaline cartilage there. The hyaline cartilage in the joints doesn't have perichondrium on the surface, so there's no perichondrium, remember, because it will be hurtful. It will hurt because perichondrium contains nerves and vessels. Okay, but we have this fibrous layer of capsule around, and we also have something strange here, and that is synovial membrane here. Synovial membrane, which is made of cells, some cells, which produce this synovial fluid. And from this fluid, the hyaline cartilage can be nourished in the process of diffusion right here. So this membrane is very important. Once again, this is a picture from hyaline cartilage on the articular cartilage. I just totally messed that up. This is the picture from joint. Here we have a hyaline cartilage right here. And we don't see perichondrium. But this is normal. It's not due to preparation and stuff. So this is joint that doesn't contain perichondrium on the surface. It's not on the surface. So this is just normal state. And here we have a picture of the synovial membrane right here. And as I said before, this membrane is very important. There are cells that are in epithelioid fashion. Once again, these are not epithelial cells, but there is epithelioid arrangement here. And they provide production, resorption, and also phagocytosis. We can see it probably better right here. This is just a scheme of outer structure of the synovial membrane. So here we have these cells right here that produce synovial fluid, and we also have some kind of these macrophage-like cells between these normal cells right here. And these macrophage-like cells, they act like macrophages, so they eat and phagocytose. So this really, the synovial membrane has also ability to produce and also to take care of the synovial fluid in the process of phagocytosing the substance and newly produce some new one. And we have also the capillaries here, and these are very important because the synovial fluid, it's not only made by the cells, the active production, but by the filtration of the blood in the capillaries. So you form fluid by filtration. And okay, I think this was the last slide. I know that this was very exhausting lecture, but I hope you liked it. And if you have some questions, which you probably have, write them down in the comment section. I will reply, and also Dr. Elhalik will respond. So once again, have a nice day whenever you're watching, and bye-bye.